Let's see, yes, I think the sound works. We are going to start the next session. So please come back into the main room and find a seat. I know it's been a bit challenging with the break and the temperature, but we're going to make the best of the time that we have. So, while I see people returning to their seats, so I will be very brief in announcing the chair of the next session, who is Dorinde Kleinegris. She has a similar Dutch background as I have, but she is currently located in Norway, where she is assistant professor at NORCE Research and the University of Bergen. Very importantly also, she is coordinator of the InnoAqua project, which is also one of the H4P projects. So I'm very happy that you will chair this session and introduce the rest of the speakers.
Thanks so much, Paul, and good to see you all here. So my pleasure is to introduce this session, and as was already mentioned earlier, the session is about sustainability, and it takes the whole width of sustainability. So it's about environmental sustainability, economic sustainability, but also social sustainability, all the way to health and for example, allergenicity. The first presentation today is by Birgir Smárason. He's a research group leader at Matís, which is an Icelandic food and biotech company, and he is an LCA expert leading research and initiatives at transforming food systems to address their urgent challenges of climate change. So, Birgir, the floor is yours. Birgir, well, Birgir is coming up. Well, while Birgir is coming up, Birgir will present about what does it take to know if an alternative protein is sustainable. Lessons from 4 years of integrated assessment.
Thank you, Dorinde, and hello everyone. So I'm Birgir, and I'm going to talk about exactly this. When I came up with the title, it's a bit lofty and grand, I think, but that was before I realized I had 7 minutes or something. So I had to make it a bit compact. But I'm going to present mainly the results and the work that we did within the GIANT LEAPS project. So I led the work package 5 on sustainability and climate. And we had the job of assessing these 9 protein sources identified within the project, and taking them through these 6 different assessment methods. And the methods were environmental life cycle assessment, using PATH, life cycle costing, looking at the economic viability across the value chains, social LCA, looking at social risks, and biodiversity and ecosystem services, looking at things such as land use, habitat pressure, climate adaptation and mitigation, looking at the exposure of the systems to climate change, and then circularity. I have to acknowledge the partners that were working on this. It was a huge amount of work, a lot of challenges, a lot of discussions, but they really did a great job. So, having said that, how did we assess? So obviously these are a lot of assessments. So it's 6 times 9, so it's a lot of work. But how do we combine them, and how do we make it make sense in the end? So we looked at the assessment methods, and for the first about 12 months of the project, we worked on combining these and trying to align into a single framework that we kind of custom made for GIANT LEAPS, the GIANT LEAPS Sustainability Assessment Framework that we have here. And just to explain it very briefly, we can see at the top where the system mapping is, where we are mapping all the production systems, and then down to the inventory data collection, collecting all the data. Then on the left side, we have the life cycle sustainability assessments, the LCA-based methods, which already complement each other pretty well. The environmental LCA, the LCC, and the social LCA. And then on the right side, we have what we call the complementary assessments, where we are looking at the biodiversity and ecosystem services, climate change adaptation and mitigation, and circularity potential. And we try to describe how they interact, which data they used from each other, and then they all feed down to these what says here at the bottom, comprehensive sustainability profiles, which we will talk about next. And the general functional unit for all of this, we worked with 1 kilo of protein at factory gate, which is called cradle to gate, and with primary data from producers across EU. Well, at least that was the dream. So how did we then communicate this complexity? So that was with the sustainability profiles, as I mentioned. So these are kind of also custom-made for the project, but I would say for European protein context. So what we did was looking at the sustainability dimensions that we discussed on the previous slide, the environmental, ecological impact, greenhouse gas emissions, land use, etc., the social foundations, working conditions, community impact, gender equality, economic feasibility, cost competitiveness, value chain viability. And then we looked at many different methods on how to do this, basing this on the absolute environmental sustainability assessment framework, the planetary boundaries, donut economics proposed by Kate Raworth, the SDGs, and our very own Venla Kitta and Hanna Tuomisto. So when we mixed all this together and calibrated this for the protein for the European context, this is what we got. And reading the profile is not that complicated. You can see the red dots and the green dots and the yellow dots. So this is the classic traffic light system. When it's green, it means it is within threshold. When it's yellow, it's approaching the limit. And when it's red, it's exceeding this threshold. The boundaries of our planet, basically what our planet can sustain. And for the social sustainability, that's a bit different and another story, but here it is expressed as highest potential social risk, which is why we have these bars and not the red and yellow light system. And I had to put just a small disclaimer at the bottom that each profile, at least which I'm showing here, is case specific, so it's not representing or reflecting a protein source or a region or a country, so that's clear. So our goal was basically to create a profile for each of the alternative proteins that we assessed. And behind each of these profiles is mountains of data, months of calculations, alignments, and different results that we had to get into this form. So it was not an easy task. So here we have the similar systems or in similar categories, more of the novel type of the alternative proteins, a microalgae and a single-cell protein. So the profiles are pretty similar trade-offs, but the energy source, I would say, because they have high energy demand, the energy source determines really whether they can reach their full potential. Another one, plant proteins, this is for oats and quinoa. The oats are produced in the northern part of the world, where the quinoa is more down south in Europe. So the profiles are maybe similar, but the social profile is really different, where the quinoa shows elevated risks, which is actually driven by upstream supply chain inputs. So this was 4 years of data collection, and we had really high hopes, but it was still not enough. And I want to first point out that the data alignment across these methods, so all the different assessments, I mean, they are very different in function and in data need even, and even system boundaries. But harmonizing them required methodological choices that are not really standardized yet, so we had to improvise a bit. And then secondly, primary data is really difficult to collect at scale. In the end, we were quite lucky in GIANT LEAPS. We had pretty good relationship with most of the partners, and in the end it worked out fine. But we also had to reach out outside of the consortium, talk to a lot of different stakeholders, and many were reluctant to share cost and supply chain information, which is understandable up to a point, definitely. So data gaps required proxy values that introduced another layer of uncertainty into these profiles. And then about scale, I mean, comparing something at a pilot scale with something that is produced at millions of tons introduces also some incompatibilities. So the profiles reflect this. So we are looking at a state that is frozen in time, so we are not projecting scale. So this represents how it is right now, or with the data that we have. So this is just another way of visualizing or putting the research into context. There are so many different ways that we can play around with this. So here at the bottom we see the greenhouse gas emission per kilogram of protein, or the carbon footprint of a kilogram of protein. Then we see the climate resilience going up, so higher is better. And we have a target zone based on the thresholds that we are working with. So we can see that only the faba bean is within this target zone, but the other plant proteins are maybe not that far away. Then we can see, this cluster here. This is the more novel tech-driven cluster with the crickets and the microalgae and the single-cell protein. So they're really high on the climate resilience, but they are a bit higher on the emission scale. But we have to consider that this was done with the data that we have, with the scale that they are currently working on, or the scale that the data that we got was. So there are many factors to consider here. And of course, the energy demand there is a bit higher, which also then is very sensitive to how we model or what we use as the energy. So you might think, this is maybe not looking so great, because you saw on the profiles that we have a lot of, red dots and yellow dots, and these are supposed to be our more sustainable options, which is maybe a bit strange. But there are a few things we have to consider, that the limits for these factors, it is very strict. So keeping within the planetary boundaries is pretty tough. But these are actually the best we got. So if we put it in context with the classical beef, it actually breaks the scale so hard that you can see, calculated for 1 kilogram of protein, it's up to 500 kilograms CO2 per kilogram protein. So all things considered, it's still doing pretty well. There are many other things to consider. I think every protein has a context where it succeeds. Every protein we assessed has these distinct sustainability profiles, with patterns of strengths and weaknesses and many different trade-offs. And I really think the science isn't about finding the best option, but understanding the trade-offs and finding the right mix. And as I mentioned, these profiles, they really set a high bar. They show that where alternative proteins need to improve, but not that the transition isn't worth making. And as I showed, red meat would simply be off this chart. And then, there are also many policy related things that we can mention, specifically related to this work. But just 3 things I want to mention. One is we need to support a portfolio. We need to diversify. We need to look at regional context and regional diversity. We need to link the protein transition to the energy transition, especially when it comes to the more novel type of proteins. And lastly, we should maybe just use sustainability profiles to direct research and development, because I think the multidimensional profiling should even be required for EU innovation funding to prevent this burden shifting and direct investment at the real bottlenecks. Yeah, that's it from me. Thank you.
Thanks so much. We'll also, for this session, have the questions at the end. So then we'll go into the next presentation. What we heard here was a presentation about looking at the sustainability for a protein crop per kilogram of protein. But now Dr. Tryfon Kekes will present us about the sustainability of and the business model of using a whole protein crop, including also its side products. Dr. Tryfon Kekes is a senior researcher at the Laboratory of Process Analysis and Design at the School of Chemical Engineering at the National Technical University of Athens. And his work primarily focuses on advancing environmental and economic sustainability through integrated methodologies. So Tryfon, the floor is yours. I'm sorry.
So hello from my side also. Very happy to be here. And I'm going to present you activities regarding the VALPRO Path European funding project. Regarding the sustainability of the VALPRO Path project, this is the older version of the presentation, but it's okay, I believe. I have it on my stick, I believe. Can I bring my USB stick?
But if you have time, I would almost say just go ahead with this one.
I can, yes. Is that okay? Yes. So what is the LCA? It's just a method to evaluate the environmental performance of a product or a system in order to take more advanced decisions and promote environmental sustainability. And similar for the life cycle costing, it is a method to calculate the total cost of a product or a product system in order to evaluate and enhance the economic sustainability of the system. So why are they important? They are important because we need to take into account both the environmental and economic sustainability of a product system in order to take correct decisions to improve the overall sustainability of a product system. Therefore, in the VALPRO path, we take into account the LCA, the LCC, and the social LCA, and evaluate the overall sustainability of the developed IPSs. We have 5 different IPSs in the VALPRO path. The first focuses on farm processing, the second on varietal selection tool, the third is regarding the intercropping of pea and faba beans, the fourth is the nutrient tracking, and the fifth is the cross-regional production of different legume-based products. Regarding the business cases that we have developed, we tried to identify the challenges and what we can enhance during the implementation of the VALPRO path. The first business case that we are going to present you regards the exploitation of the European cultivated peanuts, and the main challenges are that the peanut cultivation is still a lot underexploited and underdeveloped in Europe, and the domestic production is really limited. Additionally, over 90% of the total peanuts that are consumed in Europe are imported from non-European countries like Argentina or the USA. Therefore, it is really important to see if they are environmentally and economically sustainable to be valorized and exploited in the European Union. Therefore, in the context of VALPRO path, we have a field trial in Portugal, a small field where we cultivated peanuts, especially botanics, and we thank them for that. And we had a small trial with 200 kilograms of harvested peanuts. And then we performed a sustainability assessment of different peanut products and take into account the cultivation, the on-farm deselling, and the subsequent processing. The products that we studied were the peanut milk, the peanut butter, and the peanut oil, which are currently underexploited in the European Union. Regarding the LCA results, we can see that we have different carbon footprint among the different products, with milk having the lowest carbon footprint. Then we have the peanut butter with a carbon footprint of approximately 2.2 kilograms of carbon equivalent per kilogram of product, which is slightly higher compared to the peanut milk, mainly due to the energy-intensive processes like the roasting and the drying operations. And finally, we have the peanut oil that exhibits the highest carbon footprint, primarily driven by the extensive thermal and mechanical processing within the production line. However, we have to take into account that in this study scenario, we didn't valorize or we didn't study the valorization of the byproducts like the peanut cells, and we have done some preliminary analysis to valorize these peanut cells in order to produce bioplastics or to produce thermal energy via combustion. And we can state that we can further improve this environmental performance by approximately 25% reduction in the carbon footprint. But it's also worth noting that we have some preliminary results and we compare them with different products that are analogs to this. And we can say that the peanut products, like peanut milk, the peanut butter, and the peanut oil, can be characterized as more environmentally sustainable compared to the other counterparts like almond milk, soy milk, or average of plant-based butters and palm oil. Regarding the LCC results, we can see that based on the scenario that we studied, the peanut milk and the peanut butter are profitable, while the peanut oil is not profitable. The most profitable product is the peanut milk. Then we can say also that the peanut butter is also profitable and economically viable, while the peanut oil is not profitable. However, this scenario is constrained by the limited amount of peanuts that are produced. On the other hand, the economic performance and profitability can improve if we increase the production volumes, and we performed sensitivity analysis where we increased 10 times the production volume, and we can see that in this scenario, all the products are profitable and economically viable. The second business case that we're going to present you centers around the plant-based beverage, which also Mark presented before. So the main challenges are that the faba-based beverages in Europe are still underdeveloped, despite the region being one of the main producers of faba beans. And also there is a limit in the commercial growth of these products because they are limited by the consumer acceptance issues that are related to the flavor and the taste. Therefore, what can Valpro Paths offer? The formulation and development of a model beverage that utilizes faba protein isolate as the main ingredient, and we perform the sustainability assessment covering the whole production chain, mainly the cultivation, the protein isolate production, and the formulation and production of the final beverage. And for the protein isolate production, we selected to choose the wet fractionation method, which is the most dominant in order to produce a protein isolate. Regarding the LCA results, we can see that the total carbon footprint is approximately 0.23 kilograms of carbon equivalent per 330 ml of the beverage, with protein isolate production and the beverage production being the main hotspots across the production chain. These hotspots are attributed to the energy demand that is necessary in order to spray dry the ingredient, to produce the protein isolate, and to homogenize and heat the final beverage. Regarding the sustainability benefits that we can state that they exist regarding the faba beans, they are low water use, they are nitrogen-fixing crops, and have lower impacts compared to dairy beverages. However, there are still some room for improvement, and greater energy efficiency and optimized nutrient management can further enhance the environmental sustainability. From an economic point of view, we can see that the total cost associated for producing 1 unit of the plant-based beverage amounts to 46 cents and is mainly driven by the cost associated with the beverage production and the protein isolate production. The key cost drivers are the energy, the labor, the utilities mainly associated with the production of energy, and the packaging steps. From the economic outlook, we can see that there is a promising profitability with potential for further cost reductions if we optimize the processing value chain and optimize the packaging solutions. So to conclude with what we developed and what we studied in Valpro Paths, we can conclude that the legume-based food products are strongly sustainable and exhibit a strong sustainability potential, combining both environmental and economic benefits. The processing stage and energy consumption are the main hotspots that hinder the overall sustainability of the value chains that we studied. And also the ingredient sourcing, the packaging selection, and the process optimization can significantly improve the overall sustainability performance if they are taking and correct actions are taken into account. And finally, the European-grown legumes can offer promising opportunities in order to produce sustainable protein products and value-added food products. And the main question is, how can we further enhance the overall sustainability of the alternative proteins? First of all, we have to invest in novel and conventional technologies. For example, we stated before that the wet fractionation process was a hotspot in the plant-based model beverage. We have to adopt sustainable packaging solutions with lower environmental impacts. Additionally, an optimization in the logistics and the raw material sourcing is proven to be more environmentally and economically sustainable. It is necessary to scale up the production in order to improve the economic performance and profitability of the developed business case, and to promote the circularity and byproduct valorization in order to enhance the resource efficiency. And finally, it is important to take these actions, but you also have to continue monitoring the overall sustainability, taking into account the LCA, the LCC, and the social LCA components of the sustainability. And what measures can we take to make them? It is necessary to fund R&D and pilot projects to investigate the performance of innovative technologies. Additionally, we need to support sustainable packaging through incentives. It is necessary to strengthen the local plant-based value chains and regional production, to facilitate the industrial scale-up via eco-schemes, funding and loans and partnerships, to promote a circular bioeconomy practice and the valorization of the byproducts, like was the case for the peanut cells. And finally, to enhance the collaboration between the academia, the industry, and policymakers, and in general, all the stakeholders. So that's from my side. Thank you very much.
Thank you very much, Tryfon. So now we heard a lot about environmental and economic sustainability, and our last presenter will focus on the more social aspects of sustainability. Our last speaker of this session is Professor Clare Mills. She is a professor of food and molecular immunology at the University of Manchester and the University of Surrey. She is an internationally recognized expert in food allergy, allergy risk assessments, novel food, and food safety. So Clare, hope you can go. There you are. And Clare will talk to us about the link between health and safety and sustainability of the dietary shift. Sure.
So, thank you very much for the introduction, and a great pleasure to speak to everybody about the work we've been doing in Giant Leaps. So, just to say, obviously, I am just the work package leader. These are the guys, most of them who've done all the work, and that I'm going to speak to you about now. So, at the start of the project, what we did was a safety data gap analysis. And if you are going to have an alternative protein that goes into the market, these are the sorts of things that you have to consider and would go into, for example, if you needed a novel foods dossier assessed by EFSA or the UK ACNFP, this is what you would have to do. Information about the ingredient characterization, microbiological safety, nutritional quality, including anti-nutritional factors, toxicology, and allergenicity. And so we've done that for a whole range of the different types of foods that you've already heard others talk about we have in Giant Leaps. But what we did in this work package, and because actually we want to move away from using animal models for safety testing, it's really difficult to go from a mouse or a rat and say how that relates to a human being. And we have other tools, both in silico and in vitro tools, that can help us do that. So we focused on the toxicology, and the other thing was the allergenicity. So we've actually used a range of foods, some standard foods that we know a lot about that have certain allergenic properties, peanut, milk, soybean, and low ones like rice and maize. And then we focused on alternative proteins used particularly in the human study of Clare Gaudichon, the faba bean and the yellow chlorella, as well as proteins from other legumes and insects. So this is just to say, during the course of the project, the European Food Safety Authority actually published a couple of documents that relate to the safety assessment, slightly changing the way in which they are going about things. And this is the stepwise evidence approach for safety of GMOs. And I'm just highlighting how some of the aspects of what I'm going to talk to you about relates to the safety of toxins that could be in newly expressed proteins. And many of the precision fermented proteins are actually considered under the GMO framework because of the way they're made, as well as obviously new crops and plants, sources of foods. And that includes toxins and also allergens. And it's things like protein stability studies, pH temperature, and particularly in vitro gastrointestinal fate. So Andre talked very elegantly about the wonderful work that they've done in Infogest, which is really focused on the nutritional quality. But the food meets the body in the GI tract, and actually it's a really important part of looking at toxins and potentially allergens as to how they behave in the gastrointestinal tract and how much of that protein actually gets to your cells and into your body. And so one of the things we did was we mapped the in vitro protocols used for nutrition against those of safety and come up with a new 96 well-based plate format to try and reduce the costs of things like expensive reagents that can also take account of conditions, for example, that reflect those found in infants and in people on medication like antacids, and similarly with the intestinal conditions. And I will say, I think I'm missing a slide, never mind. And we've applied those to look at proteins from faba bean, from green chlorella and insects, and we see a gradation of differences in their digestibility, which might relate to their safety and gives an indication of safety risks, with the yellow chlorella being particularly highly digested. So another piece of work using pluripotent stem cells done by the team with Maaike van der Sande in the Netherlands, actually to develop a new model of the gut wall. So there's what goes on in the lumen, there's then what goes on in the gut wall, and these are actually enabling us to grow cells that replicate what you find in your intestine. And then in your intestine, it's actually a very immunologically active organ. If you think about what you eat every day, there's lots of protein, there's lots of bacteria, it works very well. And she's actually built this with immune cells, and I just love this. I know this is, I'm a scientist, so I love this sort of thing. These are looking down on the cells, you can see how they're forming that nice layer that you would find in the gut wall. But what's really critical, this is a cross-section through it, and this is really difficult to see. But she actually has her immune cells penetrating through that epithelial cell layer. And that shows that we're actually able to replicate what we find in vivo, because that's what your immune system does. It peeps out through the gut epithelial layer and actually samples materials. So Maaike's looked at a whole range of the GIANT LEAPS foods. So we've got our faba bean, we've got our cricket, we've got our yellow chlorella. And actually the good news is she didn't find anything that was toxic to the gut barrier. So it's a great new tool, and actually it provides really important data to reassure people that these actual foods don't have anything nasty in them. And now it's not moving. Okay. Right. Let me just make sure. Too much heat. So, as I said, EFSA also, as well as looking at the GMOs, they came up with a new framework for allergenicity risk assessment of novel foods. And actually, I'm sorry it's very small down here, but actually one of the things they're particularly focused on in the allergenicity risk assessment is, does your new protein cause a reaction in somebody who already has a food allergy? So if you have a peanut allergy and you eat faba bean, are you going to have an allergic reaction? So that's the first part of what we did, and combining in silico tools with in vitro studies. And what we showed was that in silico, if you take the proteins from faba bean and peanut, they have really relatively modest levels of sequence similarity. And if you build 3D models that we can all do with AlphaFold now, you see the bits in red are the same, and the bits in blue are the bits that are very different between them. And actually, the team in Immunology, with Yara Awad and Stefan Hasbrook, built an in vitro study to look at IgE reactivity. So if you've got people with allergies, does their IgE that they make, that's a key antibody causing allergies, does it recognize that protein? So what was very interesting was that they found that the cross-reactivity between faba bean, lentil and chickpea was high, they're more similar, but they didn't react with peanut. In contrast, and this is work that Kitty Verherx did in Utrecht quite some years ago, but we also confirmed here that the cross-reactivity between shrimp and cricket is very high. So on the basis of these data, it suggests that lentil and chickpea and faba bean will not cause reactions in people with peanut allergy, but that shrimp and cricket will cause reactions in people with shrimp allergy. So that's how we've built a tool for in vitro cross-reactivity and in silico. So then the next bit is really difficult. How do you say, if you've got a new protein, like the solein we heard about, we've never eaten solein before, is it going to be a new allergen? So predicting that is very difficult, and EFSA actually say that, so they kind of don't concentrate on it. But we've tried to develop some tools to look at this. So first of all, how does it cross the gut epithelium? If you have an inflamed epithelium, is that going to affect your ability for the allergens to proteins to actually cause an allergy? And then this is how your immune system in your gut works. And there are these amazing cells called follicular dendritic cells that hold on to protein, and they present it other parts of the immune system to go on to make an antibody. Now, if you eat food protein, you will make grams of antibody to your food protein every day, and you secrete it into your gut. In people with food allergies, those switch. They don't make IgA anymore, they start making IgE, and that's one of the key things that we've tried to look at. So how did we get on? So first of all, we've had in silico models, because if there's a particular molecule called HLA, which if you don't have an HLA that recognizes that protein, you will never make an antibody to it. And this is technology that is used in vaccine production that we've been applying here to try and predict, would our new protein stimulate an immune response? And then alongside that in Wageningen, so that was done in Sofia with Ivan, in Wageningen with Harry Wichers, Paul Voss and Paulina. One of the things they've been trying to do to validate this is, well, if we take those cells, do they present that protein? This is really technically difficult to isolate this, but they managed to make it work and shown proteins being presented by these molecules, these HLA, which helps to validate some of the in silico models. And the last bit, which was actually done by Federica Orsonego in my group, working with my close clinical collaborator, Angela Simpson in Manchester, where we've actually developed an in vitro system for what we call sensitization. So this is how B cells make the antibodies, and they're the cells that make the IgE. And what we've done and shown for the first time is that the B cells of people with food allergies are highly sensitized, and they will make IgE to a lot of our food proteins. People who are healthy doesn't do anything. So it's told us that the B cells are very different. It's also told us that we need other cells, and whether it's the gut epithelial cells that Micah developed, or it's the dendritic cells that Paul is working with, that actually they're very important to join together. So that's our next steps. So we've developed new tools, breaking new ground, but there's uncertainty in the risk assessment process. So the last part of what we did was actually looking at risk assessment to inform management. And one of the issues everybody talks about, post-market monitoring, and nobody does it or knows how to do it systematically. So one of the things we've done is put together a tool called AltProp-P, and we've been working with the patient organisations in the UK, Germany, France, and others to actually get the patients to report their reactions. And what we found in the study so far is that the most reported reactions are to legumes. And you can see here, pea, chickpea, and lentil are really high up there. Faba bean doesn't feature very much, but maybe that's because we're not eating enough of it yet. But it does indicate that the diet transition to plant-based food is likely to result in an increase in allergies to legume proteins. And lastly, often what people will do is say, oh, well, we can put a label on and warn people with food allergies. It's really important that is evidence-based, that it communicates the risk effectively to someone with a food allergy. And our patient organisation, DAAB, have run this survey in Germany, and actually what they have shown is that if you have, say, insects, and they might cause a reaction in somebody with a crustacean shellfish allergy, what our patients would like is for it to be listed on Annex 2, the Food Information Regulation. And then another tricky one, which is like milk proteins being produced by precision fermentation. Technically, they cannot be labelled as milk in the EU, but actually it's very important for these people with food allergies to be able to recognise those products as milk so that they avoid them, because they could genuinely cause somebody to have a very severe reaction. And I don't believe anybody in this whole field wants someone to have a die because they've eaten an alternative protein. So we have a raft of things that we still need to do, validating the methodology that we have developed, further refining it, but also to build on these surveys to somehow establish some kind of EU-wide surveillance. In Japan, they do this every 3 years. So maybe we can do something. And my last thing is just say, food business operators, please, please integrate safety into your thoughts early on, because often one of the reasons why it takes so long for approvals to happen is because dossiers don't have complete information. So start early. Thank you very much.
No, yeah, so I just heard that in view of time, I'm going to ask you to keep your questions and ask to the speakers in the lunch break so that we can go on to the next session. So thank you very much to our speakers again, and then I'm going to give the floor to the next one.